My name is Michel Goldman, and I'm very pleased to tell you in this short presentation the current state of play of anti-SARS-CoV-2 antibodies in the fight against COVID-19. Actually, these antiviral antibodies might be extremely interesting weapons, either to treat or prevent COVID-19. Most of these antibodies are targeting the spike protein, which is the essential membrane protein allowing the virus to enter into human cells. And as you will see, what is currently considered is that the injection of those antibodies might be helpful in selected population, either to treat or to prevent the COVID-19 disease. Really, the bottom line for the use of these antibodies is the fact that we need bridges to COVID-19 vaccines. You know, even if we are extremely optimistic, it's clear that it's not before several months that COVID-19 vaccines will be available, will be deployed in such a way that the so-called herd immunity can be reached, protecting the whole population against the pandemic. And we need bridges because there is this timing issue, but also even when vaccines will be available, there will be hurdles to their deployment. There will be issues related to their manufacturing and production at scale, in their distribution worldwide. And also, especially in our countries, as you know, vaccine hesitancy might limit the vaccine coverage that we can achieve. And also, it's extremely important to realize that just as we are not equal regarding our response to the virus, we will also respond differently as individuals to vaccines. Depending on our genetic background, on our age, our immune response will be more or less efficient. This is a very recent paper published in Nature, indeed arguing that antibodies might represent this bridge that we need, but as we will see at the end of the talk, there are still important hurdles to be overcome. I think that it's important and interesting for this audience to remind that antibodies and immunotherapy in general actually are an important strategy in the fight against infectious disease. This is not new. Just think about serotherapy 100 years ago for diphtheria. But recently, there's been a real revival with the development of several genetically engineered antibodies which allow to tackle diseases such as Ebola, such as RSV, and even very recently yellow fever. If you are interested, there have been a number of papers recently published in the New England Journal of Medicine. And interestingly, as you can see here, these monoclonal antibodies target disease for which we have either available vaccines or vaccine in development. As you know, we have now a vaccine against Ebola, and there is a new vaccine in development for respiratory syncytial virus. So this concept that antibodies might go along with vaccines in the fight against several diseases is attracting more and more interest, and this is also true for HIV. I have no time to elaborate on this. Now, coming back to COVID-19, several strategies have been used to isolate neutralizing antibodies against the virus. They could be derived from patients infected with the SARS-CoV-2 virus or patients infected with the SARS-CoV-1 virus. So those are patients who were identified, as you know, 15 years ago or so. And we have also several models which, or at least two models, which can be used, animal models, to test the usefulness of those antibodies. The point I would like to make is that when we are talking about monoclonal antibodies, they might be of different types, and I would like to draw your attention to nanobodies, which are single heavy chain antibodies, so much simpler antibodies than usual human antibodies. Those antibodies are the type that you can find in the serum of camelids, such as llama, alpacas. And their advantage when they are genetically engineered is that they are very easy to manufacture. They are quite stable, both in vitro and in vivo. And a small molecule, their tissue homing has a very favorable profile. And there are opportunities to deliver those nanobodies by other routes than the IV route, for example, sub-Q and even through inhalation. Now, when we talk about antibodies to fight the virus, I think it's very important to remind you that antibodies can be extremely efficient in neutralizing the virus, in preventing, as I showed on the first slide, the virus to penetrate into host cells, but also antibodies might have detrimental effects. Those are other type of antibodies, non-neutralizing antibodies, but which are produced against the virus, and which can actually either facilitate infection of the cells or trigger the release of cytokines by macrophagic cells. And actually, these adverse reactions induced by antibodies, this is clearly something that vaccine manufacturers are closely looking at, because among the risks associated with vaccine development, there is this risk to have antibody-dependent enhancement. I will not elaborate more on this, but just to tell you that actually one of the advantages of the monoclonal antibodies is that there you can select the antibodies with the desired neutralizing activity, so you can really make sure that your therapeutic product is not endowed with this antibody-dependent enhancement characteristic. Now, the development of these monoclonal antibodies has been quite fast. Actually, I explain you why the identification of lead antibodies has been quite quick by the fact that we could use not only models, but also materials from humans infected either with the SARS-CoV-2 or with the very similar virus, which is the SARS-CoV-1. So we have now antibodies which are under production under good manufacturing practice condition, and we will see that several of them are now entering trials. We have still to look closely at the toxicology. The first data are encouraging. A major challenge will be indeed to ensure a supply chain which allow the production, the manufacturing of those antibodies at large scale, allowing phase 3, large clinical trials, and hopefully to get regulatory approval at the end. And this will still take some time. Also, one of the leading companies in the field, Regeneron in the US, just started a phase 3 trial, and this company is claiming that indeed it might get regulatory approval by the end of this year or early next year. I would like to emphasize here that this Regeneron product is actually a cocktail of two monoclonal antibodies. Indeed, one of the concerns is to have immune escape mutations of the virus, meaning that mutation of the virus might allow the virus to escape to neutralization by antibodies. And by using two different antibodies targeting different epitopes of the same spike protein, we might certainly minimize this risk. Now, I already come close to the end of this talk, and I would like to draw your attention to the fact that those antibodies draw a lot of attention in the US. The US government is investing a lot in those treatments. In Europe, there has been less attention, which we consider as a pity. But one of the concerns is what will be the price tag of those antibodies? What will be the possible access to those antibodies worldwide. Knowing that obviously we will not use them in all infected individuals, we will have to carefully select those who will receive these antibodies. Certainly people at high risk of severe disease, and we will try to give the antibody very early on after the contamination. Obviously, healthcare workers that we would like to protect as rapidly as possible. And it might also be that some categories in the population, for example, elderly people who might not respond well to vaccine, might also be ideal candidates, either again for treatment in the early phase or for even prophylaxis after contact with infected individuals. So I would like to conclude on this slide telling you that indeed different international organizations are working to make those antibodies available worldwide, expanding their access. This is the what these organizations, IAVI and the Wellcome Trust, are doing, with the hope to save millions of lives with these biologicals. I thank you very much for your attention.